As cells differentiate, they acquire specialized functions. For decades, scientists believed this process ended with terminally differentiated cells. Committed to a particular cell fate, the ability to switch function irreversibly lost. In a major breakthrough, iPSC technology established the possibility of inducing plasticity to terminally differentiated cells. But several limitations arose, including a lengthy multi-step lab-based process, possible inefficiencies due to experimental conditions, low fidelity with which the final cell type is generated, and safety concerns, including potential cancer risk. More recently, scientists found that mature, fully differentiated somatic cells could be directly converted to another cell type by ectopic expression of specific transcription factors, without the need to acquire intermediate pluripotency. However, this technique requires viral gene transfer, an approach which, 1, requires an exhaustive laboratory procedure, and 2, carries risks, such as insertional mutagenesis, irreversibility caused by stable transduction, and increased cancer risk caused by long-term proto-oncogene expression. Therefore, it is not well suited for point-of-care applications. Thus, a non-viral tissue nanotransfection approach was developed for direct cell conversion in vivo by Professor Chandan K. Sen in collaboration with Professor James Lee. This work has been published in Nature Nanotechnology in 2017. This approach is simple, does not require elaborate laboratory procedures, and can be used as a point-of-care approach. This approach consists of 2 components. First, a hardware component, or chip, fabricated for efficient delivery of cell-specific cargo in murine model. Manufacturing of this chip has been scaled up to fit dimensions relevant to human need. Such chip has been successfully tested in preclinical porcine studies. Second, the cargo itself, which consists of a combination of different reprogramming factors. Thus, by applying a highly intense and focused electric field through arrayed nanochannels, this device manoperates the adjacent tissue cell membranes and electrophoretically drives reprogramming factors into the cells. This new nano-electroporation technology is distinctly different from bulk electroporation. The simplicity and utility of this approach was demonstrated by nano-electroporating the skin that served as an agricultural land to cultivate cells of different functional abilities. Using a specific cargo, a combination of different reprogramming factors, skin fibroblasts were successfully converted to endothelial cells that ultimately formed blood vessels with 7 days. These blood vessel networks anastomosed with the parent circulatory system and restored tissue and limb perfusion in 2 murine models of injury-induced ischemia. Similarly, using a different cargo, skin fibroblasts were converted to neuronal cells that showed similar gene expression profile and electrophysiological function to that of cells in the developing brain. This tissue nanotransfection platform technology has the potential to ultimately enable the use of patients' own tissue as a prolific immunosurveilled bioreactor to produce autologous cells that can resolve conditions locally or distally upon harvesting. This simple-to-implement nano-electroporation technology generates favorable biological responses. It is a one-time treatment, applied topically, and only lasts for a fraction of a second, without elaborate laboratory procedures. And it could have applications beyond reprogramming-based cell therapies, such as gene, protein, and drug delivery.